Now to introduce today's speaker, Shae Swanepoel. Shae, I'm just passing the screen to you. If you could accept the invitation to present. All right. Now, Shae is a PhD student in the Department of Biochemistry, Genetics, and Microbiology at the University of Pretoria in South Africa, where she is investigating the disease resistance potential of eucalyptus. She will be presenting research she did during her master's on the molecular dialogue between Eucalyptus grandis and the myrtle rust pathogen. Shae, if you're all good there, feel free to get underway.
Thank you, Jenny. So as Jenny just mentioned, my name is Shae Swanepoel, and I am a PhD student at the University of Pretoria. But today I'll be talking to you about my master's research, which investigated the interactions between Eucalyptus grandis and the pandemic biotype of myrtle rust. This work was done in collaboration with quite a few people, but notably Louise Shuey and Jeff Pegg, based in Australia, as well as then Prof. Siniska Naidu, who's based at the University of Pretoria. So I wanted to start off to give a bit of context about where I'm from. So I'm at the University of Pretoria, and I'm based in the Forestry and Agricultural Biotechnology Institute. And this is quite a large institute. It consists of about 240 people, more than 20 academic staff. There's a whole host of postgraduate students, postdoctoral fellows, research visitors. And then interestingly, we have approximately 30 languages are spoken by members of FABI. And I think this just showcases the diversity within our institute. But then part of FABI is the Eucalyptus and Pine Pathogen Interactions Research Group. And this is the group that I'm a part of, led by Professor Siniska Naidu. And I've just attached quite an old picture here of us. There's a few more members that have joined in the recent years, but this is just a nice visual of who we are. And then if you would like to go and see who our FABI website, the FABI website has our profile on it. You can scan the QR code here to go and see what we do. So again, just a little bit more about us. We focus on the interactions of pests and pathogens with eucalyptus and pine. I specifically work on eucalyptus, and the main focus that we've had in eucalyptus over the last few years is focusing on myrtle rust. We look at the stem canker pathogen that causes these hideous galls or cankers on the stems of eucalyptus. We look at the Lectocybe invasive, a blue gum chalcid, root rot caused by Phytophthora cinnamomi, and then we also have Teratosphaeria leaf blight. And our focus has been more on the transcriptomic sides of the interactions between pests and pathogens and eucalyptus. But let's move on to what we are here for today, is to talk about myrtle rust. So I wanted to talk about myrtle rust in South Africa because I think many may not know what the situation of myrtle rust is in South Africa. And so it was first discovered on a non-native ornamental Myrtaceous communis tree in 2013 by Yolanda Roux. And it was found to have a rather broad distribution across South Africa. I think that the distribution was about 1,500 kilometers. But it is interestingly a unique genotype that is distinct from the pandemic biotype that we see in Australia or New Zealand. And they hypothesized that it was a single introduction from a still unknown source. And what is most interesting about this is that it is less virulent than the pandemic biotype that is seen in places like Australia. And I think to date, we still don't really know why. And also, there have been no reports of this particular biotype or genotype posing significant threats to our native or introduced Myrtaceae species. We do have a number of ornamental Myrtaceae species, and that can include Eugenia, Syzygium, and whatnot. But we also have many industries that are cultivating non-native Myrtaceae species like eucalyptus, and this includes our paper industries like Sapien Mondi. And so this really highlights that we need to focus on protecting not only our native species, but our introduced species as well, because there's always the threat of reintroduction of either the pandemic biotype or our biotype becoming more virulent. And so considering this threat, we developed a study to identify mechanisms that contribute to host defense in Eucalyptus grandis, as well as pathogen virulence in the pandemic strain of myrtle rust, to hopefully identify resistance mechanisms in eucalyptus, as well as targets for control for this pathogen. So our samples were obtained along the east coast of Australia, our eucalyptus samples. And this is interesting because it means that they're not clones. They are a mixed provenances, and they come from the complete geographical range along the coast of Australia. And this ranges from Coffs Harbour to North Queensland. And the seeds were collected and supplied by Dr. Simon Southerton, and Louise Shuey then worked on that side to perform the experimental studies to get the data that I would look at. So she did a fantastic job. She identified resistant and susceptible Eucalyptus grandis across that natural range. And then in collaboration with Jeff, they were able to perform inoculations and disease scoring to identify these hosts for our study. And so the disease scoring was rated on a scale of 1 to 5, where 1 represents resistant hosts and 5 represents susceptible hosts. Resistant hosts, as you can see from these images, had no pustules, and there were no symptoms of disease or chlorotic lesions. And the susceptible host had pustules that were greater than 1.6 millimeters in size and more than 20 uridemia on the leaves, petioles, and shoots, and they were just very diseased. So for this study, we selected the extremes of resistance and susceptibility. So we only looked at 1 and 5 to better understand what contributes to these extreme phenotypes. But it's needless to say that ratings 2 to 4 are still important, and future work can actually look at what the spectrum of disease resistance is. So what we ended up doing, as I say, we selected resistant and susceptible samples. We had a time series, which is quite unique to studies on myrtle rust and eucalyptus. And then we performed RNA sequencing and a dual mapping of our reads to the host and the pathogen for these ratings 1 to 5. So I wanted to just briefly talk about what dual RNA sequencing is and how it differs from standard sequencing. So the biological sample that contains both host and pathogen transcripts would be ground down, and then total RNA will be extracted. So this differs because you're including both host and pathogen. And then you perform the standard RNA sequencing, where we have the library preparation, the sequencing here, and how this differs from standard sequencing is that you sequence to a very deep read depth. So we sequence to about 40 million reads, which would enable then the identification of both host and pathogen transcripts. And then you perform dual mapping to a concatenated genome for the host and the pathogen, and then you can identify which transcripts derived from which organism and perform the typical downstream analysis, such as expression quantification and differential expression analysis. So that's what we did. And this is the first paper that we published on this work. And it implicated brassinosteroid signaling in defense against myrtle rust. So I'm not going to speak too much to this image, but what I wanted to show here was that we found extensive transcriptional reprogramming of the resistant responses at 2 days, and particularly at 2 days post-inoculation was only mimicked in the susceptible hosts at 5 days post-inoculation. So we see a lot more transcripts or differentially expressed genes rather in the resistant hosts at 2 days post-inoculation, which is only really mimicked at 5 days post-inoculation here. So I neglected to mention that this is showing differentially expressed genes. So those are genes that are then induced significantly upon the pathogen infection. And so we'd be interested to know what these genes are, and this is exactly what we did. We performed gene ontology enrichment to see which pathways were significantly enriched among these differentially expressed genes, because this would give insights into the mechanisms that the hosts are employing against this pathogen. And one of the interesting things we see across all of our gene ontology terms pretty much is that for the resistant and the susceptible hosts do have the responses that the resistant hosts have, but they tend to be very delayed. So the enrichment of these terms only happens at around 5 days post-inoculation. For example, this plant type hypersensitive response, which would be the response that causes the programmed cell death and the localized cell death to prevent proliferation of the pathogen. So while these terms are enriched earlier in the interactions in the resistant host, in the susceptible host, they are only induced later. And then when we look at which phytohormones are enriched, this is where we start to see quite interesting patterns. So brassinosteroid mediated signaling popped up as an interesting term within the gene ontology enrichment. And this was at 2 days post-inoculation in the resistant host, whereas there were no enrichment of brassinosteroid mediated signaling terms in the susceptible hosts. And so this means that potentially this pathway, or the genes that underlie this pathway, could be contributing to disease resistance. Notably as well, we had a lot of salicylic acid mediated signaling, but you can see again that these terms are absent in the susceptible host at 1 and 2 days post-inoculation. So while these terms do exist within the data, we can see that the susceptible hosts tend to have sort of dysregulation of these processes, and that could potentially be causing this susceptible phenotype that we are seeing. And so we dug then a little deeper into brassinosteroids and brassinosteroid-mediated signaling to see which genes were underlying those terms. And from literature, we know that in some instances, the application of brassinosteroids, the exogenous application of brassinosteroids, can lead to reduced disease symptoms against biotrophic pathogens, which myrtle rust is a biotroph. So there is some connection here between brassinosteroids and then the resistance of eucalyptus against myrtle rust. And at 2 days post-inoculation, where we did see quite a number of differentially expressed genes, we have this pattern of upregulated brassinosteroid-mediated signaling associated genes. And this pattern is not seen at 2 days post-inoculation in the susceptible hosts. And only really do we start to see an upregulation at 5 days post-inoculation compared to the resistant host. So that pattern is only really seen quite later on in the interaction. And again, this comes down to the hypothesis that these susceptible trees do have the mechanisms to defend against the pathogen, but they are just delayed. And it's that delay then that could potentially be what is causing the susceptibility phenotype, and these plants cannot succumb to, or cannot prevent disease proliferation. So following this, we then wanted to look at the resistance, the myrtle rust resistance QTLs. And we wanted to know which genes firstly underlie these QTLs, and then what is the expression of these genes in our data sets that underlie these QTLs. So 5 of these have been identified to date in eucalyptus. So we have the first one, which was identified in 2003, and then we have 4 more that were identified in 2016. And so we wanted to know then, investigate these a little bit further. So we looked at the total number of genes that were underlying these loci, and then we wanted to look at what the expression of these were, to highlight potential targets for improved breeding against myrtle rust disease. So if we look at the first one, the pathogen, the Puccinia psidii resistance 1, that was by Younghans et al. In 2003. And we found about 382 genes that underlie this loci on linkage group 3, so essentially chromosome 3. And then the additional ones, we found 459 that underlay Puccinia psidii resistance 2. 752 were underneath PPR3, 506 were PPR4, and then we found 941 under PPR5. And then when we investigated the differential expression patterns, you can see here that there were quite a few of these that were differentially regulated upon their infection with this pathogen. And interestingly enough, 2 genes that were underlying Puccinia psidii resistance 3 and 4 underlying PPR5 are actually involved in brassinosteroid signaling. So we wanted to know, or we still want to know rather, what these genes are potentially doing in this interaction, and if they are contributing to the brassinosteroid signaling that we're seeing in the resistant hosts. So then we moved on to looking at the resistance genes that were under these QTLs. And we can see that there's quite extensive transcriptional programming of these resistance genes. And we found quite a number of them that were differentially regulated. There's no real clear pattern here. We can see that PPR3 is significantly upregulated upon infection, whereas PPR2 maybe not so much. But what is important is that we have these lists of genes, and we can then further study these to see what are the roles of these genes in this interaction, functionally characterize them, and potentially start implementing them in improved breeding programs, for example. So then we wanted to move on to the pathogen responses, and this is the second paper that we published on this work. It's the implanted gene expression of Austropuccinia psidii in resistant and susceptible Eucalyptus grandis. So while we had really good data for the hosts, the pathogen was a bit of a different story. So I show the mapping statistics here and the total number of expressed myrtle rust genes over the time series. And you can see from these dashed pink, reddish, and blue lines that the percentage of reads that mapped to the host, to the myrtle rust reference genome, were quite low. And we can only see about 0.6% that actually mapped to the reference genome for myrtle rust. And this was at 5 days post-inoculation in a susceptible host. So what this may mean is that we're not really getting the full picture of what is happening in the pathogen during this interaction. But having said that, we did still find a number of not differentially expressed genes, a number of total expressed genes, about 890, and about 888 of those was actually seen in the susceptible host at 5 days post-inoculation. So it may not be the full representation of what is happening in myrtle rust during this interaction, but it's still pertinent to go through this data to see what it is that we can fish from it. So we then used the putative effector genes that Perry Tobias identified in her genome paper. And we can see that a few of our genes, of these 890 genes that were expressed, a few of these may actually be putative effector proteins. So in figure A, we can see that the pathogen effector gene was either expressed in the resistant host, represented by this blue, either in the susceptible host, represented by the pink, or they were shared in both, or they were expressed in both the pink, in both the susceptible and the resistant hosts. So when we looked at what these genes were, many of them did not have any annotation. So this means that we can't really decipher what they are doing in the interactions, but we do highlight them as candidates for further functional testing. And interestingly, there were a number of genes that were uniquely expressed in the susceptible host at 5 days post-inoculation. And the one that I'm particularly interested in is this rust transferred protein, or RTP1. And when looking through literature to see what this is, in a study by Kim et al. In 2013, they found that this protein localized to the extrahistorial matrix during early stages of infection, which is, we can see by this image on the left. And it also localized inside the host cell cytoplasm as the disease progresses and the historia matures. So they postulate that this protein is important in maintaining the biotrophic lifestyle of the pathogen. And then as the Historia matures, more concentrations or higher concentrations of this protein can be found in the cytoplasm, which you can see here in the right-hand side. And so as a result, cyclosis, which is the movement of cytoplasm within the cell, is inhibited. And this then means that the movement of the host nucleus and the chloroplast are also inhibited. And so from literature, we do know that the chloroplast does play an important role in defense. And accumulation of the chloroplast near the Historia causes these shadows to form. And then this thereby reduces the photosynthetic activity and reduction in the chloroplast-derived reactive oxygen species. So ROS production is reduced. And then the host cell is less able to defend against the invading pathogen. And then also by the rust transferred protein that then starts to accumulate near the Historia, it prevents the Historia from degradation. And then this is what is actually potentially extending the biotrophic phase of the pathogen. And so by seeing this protein in 5 days post-inoculation in the susceptible hosts, we can say that it probably is it is occurring earlier in the interaction. We are just not seeing it in our transcriptome data. But at 5 days post-inoculation, specifically in the susceptible host, the expression of this gene is so high because of this accumulation that it may actually be playing a role in this disease susceptibility. And so this is quite an interesting protein that I think needs to be studied a little bit further in our pathosystem. And so we were not able to perform gene ontology enrichment on the differentially expressed genes because we found very few of them, if you consider that we only had about 890 total expressed genes. So we ended up performing gene ontology enrichment on the total expressed genes. And from this, we saw that many basal pathways were identified. What I mean by that is pathways that the pathogen potentially needs just to survive, not actually cause disease. And these can be also further investigated as housekeeping. We can find housekeeping genes that can then be used for future control methods of this pathogen. But one of the interesting pathways that we did see was those that are involved in oxaloacetate production. And most of the data you can see from this figure sits with the susceptible hosts. So is then oxalic acid potentially involved in myrtle rust virulence? So oxaloacetate is produced by the reduction of malates by malate dehydrogenase, and oxaloacetate is a precursor for oxalic acid. And so we know from studies in chestnuts that Cryphonectria does use oxalic acid to cause disease. And so is it possible then that myrtle rust is utilizing the same technique to do that? And when we look at what the effects of oxalic acid are on the plants, it influences the production of phytohormones. This is predominantly by affecting, for example, the downstream signaling responses of salicylic acid. And then, like I said, it affects the downstream activation of defense-related genes, but it also reduces plant reactive oxygen species. And from our host data, we can see that the susceptible hosts can't really regulate the production of reactive oxygen species. And so is this potentially what the pathogen is utilizing to manipulate that pathway? And when the reactive oxygen species is the production thereof is affected, then localized host cell death is inhibited. And when you inhibit the host cell death, the pathogen can't proliferate any further, and this then contains the disease. And so when this is affected, potentially then that localized cell death is not occurring and the pathogen does proliferate. And then this leads to a spread of disease. So then to conclude this research, I wanted to introduce a model of plant defense. So when pathogen recognition receptors, these proteins that sit on the intracellular membrane between the outside and inside of the cell, when they recognize pathogen-associated or microbe-associated patterns, which are these pathogen-derived molecules, it leads to a cascade of downstream activation of hormone signaling, which then leads to secondary metabolite production, activation of transcription factors that then lead to the expression of pathogenesis-related genes, which are the classic defense-related genes. And then it's these genes that produce proteins that have antibacterial properties, antifungal properties, and then they also have a role to play in cell wall reinforcement. Similarly, when our genes recognize these effector proteins, it also leads to a similar cascade of events, but we also see hypersensitive responses that are caused by the accumulation of reactive oxygen species. So what we are thinking is that brassinosteroids, while typically not known to be a defense-related hormone, has been shown in recent years that it does play some role in disease resistance or in the defense responses of plants. And so we are thinking that brassinosteroid signaling enhances the defense responses of eucalyptus, somehow by potentially manipulating salicylic acid, because there has been literature to show that there is an interplay or a crosstalk between these hormones. And then potentially this pathogen-derived oxalic acid may then act to reduce host defense responses. And this could be by the reduction of the production of reactive oxygen species, as well as influencing hormone signaling. But what effect this has downstream is still not something that we know. And future research does need to focus on this pathway or these pathways as a whole to determine what is actually happening in this interaction. And so we envision future research to investigate the effects of brassinosteroids on eucalyptus defenses. So as I've mentioned throughout the talk, brassinosteroids have been known to contribute to the defense responses of other plant species. So we want to look at applying exogenously brassinosteroids to eucalyptus and then measuring the effects that this has on fungal growth. Does it influence the pathogen more than the host? We can also then look at phytohormone levels. So we can determine if brassinosteroids application is influencing the accumulation of other phytohormones and thereby the signaling of other phytohormones. And we can also then look at the defense gene activity. So if you remember the model that I previously showed, those pathogenesis-related genes are very important in activating the defense responses of plants, which includes those antifungal, antibacterial properties. And so when brassinosteroids are applied, we can then look to actually measure the expression of these genes and see, does the expression of these genes change in the presence of this hormone? But of course, as I'm sure you know, we cannot study the pandemic strain in South Africa because it is a quarantine threat. And so any future research would need to be performed that side of the world. And we are always looking for people to potentially collaborate with, because this is something that we would like. That I would like to then thank the following people. So Louise Shuey, she had the experimental design, she performed the inoculation, she did the RNA extraction, she sent the samples for sequencing. She did a fantastic job, and I was able to analyze the data that she generated to such a high standard. And then Jeff, he assisted Louise in the inoculation trials, and he provided insights into our data. We met a couple of times to discuss what our data could potentially mean. And then to Simon Sutherton for providing Louise and Jeff with the seedlings that we used for this study. And then also Sanushka Naidu for conceiving the study and then the support. And then my email is here, so if you'd like to pop me an email with any questions, then please do. Thank you.
Thank you so much, Shae. That was really well done and really fascinating. I'm
Thank you,
happy to
Jenny.
field questions from the audience. Audience, if you can pop up questions into the chat, that would be great to have some questions for Shae. One question that came through is, could you knock out brassinosteroids and see the effects on the plants?
That's a good question. I think because the brassinosteroid pathway is so vast, it may
Hmm.
be difficult to knock it out because it would be difficult to figure out where in the pathway the genes are playing that role. So I think we could either do exogenous applications, but we could also look to do um, apply. I know there are some chemicals that you can apply that inhibits the pathway. So you can't just necessarily knock it out, but you can
Right,
inhibit the
right.
pathway and then see what the effects are of the inhibition and the application. You could potentially do that in tandem.
Yeah, interesting. And then do you have any ideas for how you would investigate those 2 through 4 rated leaves? Would you use the same method that you've used here, or would you use a different method or vary the method somehow?
Also a good question. I think it's a difficult one because we got really good data from the host by doing this method, but we weren't able to really fish apart the pathogen responses. And I think that is because, for example, you're taking the whole leaf and subjecting the whole leaf to RNA sequencing. And so you're somewhat, I want to say, diluting the pathogen transcripts within the host material. So when you're looking at more of the resistant like 2 or 3, which is the moderate, 4 is sort of moderate as well, you may not see the responses that you would like because there won't be as many pathogen derived transcripts within the plant. So it's a bit of a difficult one. I think potentially we need to focus on actually honing in that sampling method and that RNA sequencing method, maybe doing more of a targeted approach if we identify leaf discs that have more pathogen, per area,
Hmm,
then we could possibly see more of the pathogen than we do when we have the whole leaf, for example.
interesting.
Yeah.
Another question's come in. Oxalic acid is usually associated with necrotrophic pathogens. Are you aware of
Yeah.
any other biotrophs that secrete oxalate to suppress host defenses?
Yeah, so that is true. I'm not sure on the literature that stands on oxalic acid and necrotrophs. So I'm not too confident to answer that question, but I do think that be, it is something that we do need to look really deeply into with this data and with this pathosystem because it could not be. Um, it not enough research is done there, and with, like I say, the data that we had, we didn't really get the full picture. So yeah, I think more research needs to actually go into seeing if oxalic acid does play that role.
Speaking of more research, you said at the end that you can't really study myrtle rust, or at least not the strain that makes the biggest
Yeah.
difference in South Africa. Are you continuing with myrtle rust with your PhD, or are you working on something else right now?
Yeah, so as I say, we do have that strain here. We do have our own unique biotype, genotype strain of myrtle rust here, but it's not causing the issues that we see in other parts of the world. So not a lot of research has gone into that, which I think is a little disappointing because it would be really interesting. I'm interested to know what is the difference between these two strains and why is one more virulent than the other. Um, but in terms of my PhD research, no, I'm not looking further into myrtle rust, unfortunately. For the PhD, over the course of the last few years, we have generated a lot of transcriptomic data. And so what we are looking at is more of a combined defense response of eucalyptus to these different pathogens to see what is the sort of core defenses of eucalyptus, if you would say that. So that's more what my PhD focuses on. But I have a lot of ideas for what we can do with myrtle rice, but because we have the strain that doesn't cause problems, yeah, we can't really pursue that research here.
I have another question that's come in. If the hypersensitivity response is upregulated in resistant hosts, would you not expect to see more symptoms of the response in resistant hosts?
Yeah, so I assume that they're talking about the leaf where there weren't any of those chlorotic lesions. That's a good question, and that is something I think that confused me a little bit as well. Um, and I have to go back and look at the the data to actually answer that question. Yeah, I'm sorry. It's been a while. Yeah, I have to go back and look at that because that is something that came up when we were doing the research, and we did have a hypothesis for it, but it evades me now. I'm so sorry.
That's all good. All right. I think that's it. Thank you so much, Shae. This was a really great, really informative talk, and you have a couple of compliments here, but I'll send those through to you separately.
Thank you so much.
So, yeah. Thanks again for your presentation. Okay, a video of today's webinar will be made available on the Beyond Myrtle Rust website in the next couple of days. It will also be emailed to everyone who registered. This has been our last webinar of the year, so thank you, Shae, for taking the time with us this evening / this morning, your time. We hope to continue to bring you the latest in myrtle rust research next year and are actively exploring funding options to keep this series going. If successful, our next webinar will be sometime mid-February. So everyone, keep a lookout in your invite for the invite in your inbox next year. Thanks, everyone. Harira, hambakashle, tot ziens, and see you next time.